Hello, everyone, and thanks for joining us. I'm Dave Iverson, contributing editor at the Michael J. Fox Foundation and the moderator for our Third Thursday series of webinars. Today, we're going to focus on the year in review, all of the things that have taken place in terms of Parkinson's research during the year 2016. And it does mark the end of our Third Thursday webinar series for the year. We thought we'd pass along a little bit of information about how many people have participated over the course of the year. More than 22,000 people have watched at least one of the webinars during our Third Thursday series, either live on the third Thursday of the month or through our on-demand offerings. And we really just want to take a moment to thank all of you for participating, both those of you with Parkinson's, those who have family members with Parkinson's, clinicians, researchers. Together you make up our Parkinson's community, and we're really grateful for your participation in this series. It's something the Foundation really values, and so thank you for joining us throughout the year. We have a lot to cover today in this year of review, which is a good thing in this program, rather, about the year in review, because so many interesting things have happened during 2016. And so because of that, we're going to do an extended webinar today. Instead of our usual 60 minutes, we will be going for a full 90 minutes. So we'll go until 1:30 Eastern, 12:30 Central, 11:30 Mountain, and 10:30 on the Pacific Coast. You'll be able to submit questions, as always. Just use that Q&A box that you see in the middle of the screen. You can type your questions in there, and we'll do our best to get to as many as we can, and hopefully we'll get to an additional number than is our norm, given that we're going for the full 90 minutes today. Last, before we get started, as always, our slides are available for download. There's a box called Resource Lists on your screen, and you can click there, and the documents will open an additional browser window, and you can save or print from there. All right, let's get started with what we're going to be talking about in our webinar today. We're going to focus initially on what progress is being made in terms of developing a therapy that would actually slow down or stop Parkinson's disease. We'll also talk about new symptomatic treatments that are either now on the market or getting close to the market. We spend a fair amount of time talking about what we're learning about the underlying biology and the cellular connections, which we think are really the key to solving the mystery of Parkinson's disease. We'll also spend some time talking about new progress in terms of being able to come up with a more objective and definitive test for Parkinson's disease, and about the need to make further progress with finding objective biomarkers for the disease. And then we'll wrap up by talking about some new updates with things regarding things like deep brain stimulation, stem cells, and also the latest on exercise. All right, here is who is joining us for our session today. First off, Gary Rafaloff. Gary is from Marlborough, New Jersey. He was diagnosed with Parkinson's back in 2012 at the age of 60. He's been extraordinarily active in participating in our Parkinson's community on a number of levels, including participating in a number of clinical trials for new Parkinson's therapies. So Gary, thanks so much for being part of our webinar today.
My pleasure. Happy to be here.
Happy to have you. And joining us as well from the Michael J. Fox Foundation is Dr. Rachel Dolan. Rachel is the Vice President of Medical Communications and also is a trained movement disorder specialist. Rachel, welcome. Thanks for being part of our program.
Thanks for having me.
And here, too, is Dr. Todd Scherer. Todd, of course, is the CEO of the Michael J. Fox Foundation. Todd, thanks as always.
Hi, Dave.
All right, let's begin by beginning our discussion about new therapies to slow or stop Parkinson's disease. And you see at the heading of this first slide that we're going to focus first on what we're calling repurposed therapies. These are actually drugs often that have been approved to treat a condition other than Parkinson's, but now may be showing some promise as possibilities for Parkinson's. And before we go through some of these individually, Todd, just take us a moment, for example, if you would, and tell us a little bit about what's so intriguing about these repurposed therapies, the advantages that they may potentially have in terms of how quickly they might become available.
So the main advantage of working on a repurposed drug lies in the fact that these are drugs that are already FDA approved or available for other reasons or to treat other diseases. Why that's exciting is that means that the drugs have passed through a lot of the hurdles and roadblocks that normally derail or delay the development of a new drug. For example, they've been shown in certain, in populations to be safe and effective to treat different diseases. And for that reason, a lot of times you're able to jump over a lot of the steps, particularly some of the preclinical testing that is needed to be done when it's a new indication, a new drug, in order to immediately start testing the drug. The other advantage is once you get a nice building of evidence to support the drug for the treatment in Parkinson's, it'd be much more accelerated for it to be available for people because the drug is already on the market. I just would like to add, though, that with all those advantages, we still need to do the same type of rigorous evaluation, sometimes, for example, for the safety of the drug. So while the drug that's available may be safe for a certain disease, it doesn't mean that it has been tested in Parkinson's, and there we do need to really evaluate that safety. And certainly we want to make sure we're establishing the efficacy of the drug before we would be comfortable exposing large numbers of people to the drug as well.
Well, by way of example, Todd, then tell us something more about the first one we see on this list, is Ratapine, which is a hypertension drug. And so it's a drug that's used to deal with people who struggle with high blood pressure. And this seems to me an example of the kind of thing you're talking about, because, of course, in Parkinson's, many people have the opposite problem. They have low blood pressure. So I assume one of the things that's being tested now and is probably already on our way to determining is whether or not this drug, which is designed to bring down blood pressure in people who have hypertension, whether it would bring blood pressure down too far in someone with Parkinson's disease who already might have lower blood pressure. What have we found out about that? And then say something more, if you would, about the promise of this drug and why scientists are interested in how it could have an effect on Parkinson's disease progression.
Yeah, so first, to just talk about why we're interested in this drug in the first place for Parkinson's, because it may not be obvious to many people why a drug that was developed to treat blood pressure could be of interest for Parkinson's. There's similar biological processes that are used to control the functions across different bodily systems, and in this case, in control of cardiac function, that is regulated in the body by regulating the amount of calcium that gets in and out of the muscle that is the heart itself, which is the muscle. So by the calcium getting into the cell and out of the cell is sort of how we regulate the heart beating and function of the smooth muscle in the heart. Interestingly, the activity of neurons in the brain is also regulated by the influx of calcium into the cell and also getting it at really tightly controlling the amount of calcium as it gets in and out of the cell. And what an investigator at Northwestern had found many years ago is that dysregulation of calcium levels in and out of the cell was contributing to the degeneration of the nigrostriatal neurons in the brain cells affected in Parkinson's. And in fact, by adding an inhibitor, a calcium channel inhibitor, which is what Ratapine is, you could increase the survival of the brain cells impacted in Parkinson's. So that led him to sort of explore the fact that we had a lot of calcium channel blockers on the market already that were available for treating high blood pressure and cardiac function. And one that they identified was Ratapine, which was a particular of interest because it also had the capability of getting into the brain. So this is sort of one of the other aspects that we need to look at in terms of repositioning is because a drug was developed to treat disease in the periphery, like cardiovascular disease, doesn't mean that drug actually has the capability to treat a neurological disease, and for example, needing the ability to get into the brain. There also wound up being additional epidemiological studies looking across populations that found that people that were on antihypertensive drugs like Ratapine actually had a decreased risk of getting Parkinson's. So based on both the laboratory data and then this epidemiological data, led the investigative team to then explore isradipine in clinical testing for Parkinson's. As Dave mentioned, one of the things that was looked at very early in clinical testing, if you look at the slide, sort of in the phase 1 and phase 2 level areas, was both the safety, can we give this drug now to Parkinson's patients, and what would happen to their blood pressure or any of the other potential side effects, but also trying to look at the dose of the drug that we would want to explore in people. So this is another aspect that needs to be looked at in repositioned drugs. So drugs might be designed to take at a very certain dose for one indication, and we have to see, are we in the same dose range when we apply it to a disease like Parkinson's, or are we going to need 100-fold of that dose to have an efficacious effect, where you might now uncover additional safety issues that aren't relevant in the use of the drug at the lower dose. So in the phase 1 and phase 2, the researchers both looked at safety and looked at dosing and how high could we push the dose in people with Parkinson's, and we were still in the range of where we think it would be safe, and in the range where the laboratory data suggested that we could get a benefit on protecting the brain cells. So based on that data, obviously being successful, the project has now launched into the phase 3 testing, which is now really looking at the doses that we believe are tolerable and safe in people with Parkinson's. Can we get an impact on the disease progression or the symptoms of the disease in a larger study where now they are studying about 400 Parkinson's patients in the phase 3. And we should expect
Todd,
those. Sorry, Dave, go ahead.
No, I was just going to ask where I think you were going, which is how long will it be before we get the results from that phase 3 trial?
Yeah, so my understanding is that they have completed the recruitment in the study, and as this is a study to look at disease progression, it's probably evaluating the patients for a couple of years. So I would expect now that they finished the recruitment that the ultimate results from that should be in a couple of years, the outcome. Because again, we're trying to see, did it slow the trajectory of the disease over a couple of year period. So we do need to evaluate the individuals in the study for that period of time.
And Gary Refloff, why don't we ask you to make a comment at this point too, because Todd just described this, sometimes rather elongated clinical trial process, and we've already gotten a number of questions from people asking about how can they participate in clinical trials if they live in an isolated area in particular. Sounds like recruitment is finished for this particular trial, isradipine, but some of these others, recruitment is still going to be going on that we're going to be talking about in a moment. As someone who's participated in lots of trials, what's your advice on how people can best find a trial that fits their particular circumstance and in some cases, a more isolated location?
Well, without trying to make this a recommendation for our sponsor, the Fox Foundation, I have to make a recommendation that the best way is through the Fox Trial Finder. Clearly, it's the one avenue that makes it very easy to see every study that's happening for Parkinson's disease in the country, and then it could be filtered down by any patient's desires, whether they want an interventional trial, an observational trial, a symptomatic drug. They can put in the range of territory that they're willing to go to. And then once they set up their account, they will be notified whenever there's a trial opening in the territory that they live in that reaches the parameters of the type of study that they're willing to participate in. And just to divert back to isradipine, the Fox Trial Finder ambassadors were involved in that study in helping to recruit patients that we had identified as members of the Fox Trial Finder, and we were able to cut the recruitment time dramatically from that which was set in the protocol. So getting to the frustration of listening to Todd say that it's going to be a couple of years to get the results, the one way we can shorten the time period of any study is for us to sign up as quick as possible and cut the recruitment time. And using Fox Trial Finder is a great way for us to keep on top of that and sign up and volunteer for studies as soon as we see them get posted.
Great. And again, the way to sign up for Fox Trial Finder, if you're interested in participating in these clinical trials, and as Gary suggests, is probably the greatest contribution any of us living with Parkinson's or who have family members with Parkinson's, because we always need controls as well, can make, is to participate in trials. So visit foxtrialfinder.org if you're interested in doing that. Let's move along and touch on a couple of these other interesting new possibilities. And Dr. Rachel Dolan, let me ask you to comment on Inosine, which is the next one. This is something that boosts urate levels. And again, as Todd was mentioning with Exenatide, we've learned through epidemiological observations, among other things, that people with higher urate levels seem to have a different rate of either getting Parkinson's or progressing with Parkinson's. Where are we with learning whether or not Inosine may be something that could make a real difference in the lives of people with Parkinson's?
Yeah, so as you said, by looking at epidemiological studies or large populations of people, we found that Inosine, which raises urate, which is an antioxidant, people who had higher levels of urate had lower levels of Parkinson's disease. And then also among people who already had Parkinson's disease, those who have higher levels of urate showed a slower rate of progression. So that led scientists to think about this a little bit further and study Inosine, which raises urate, further. So this already went through, phase 1 and phase 2 trials, which showed that it was safe and tolerable in people with Parkinson's. And so now this is recruiting for phase 3 trials. So this will be a multi-center study across the United States, which will recruit hundreds of people to study Inosine to show if it truly is effective to slow the progression of disease in Parkinson's. And as Todd said, with Exenatide, this will be a trial over a couple of years to see if it truly does change the course of disease in Parkinson's.
And Rachel, while I have you, why don't you go ahead? I'm going to jump over diabetes and come back to that in a moment, but ask you, since you're also a clinician, to comment on nicotine, which is probably the most counterintuitive substance that's on our list here. What do we know about the connection between nicotine and Parkinson's, and how is that being tested, since obviously I'm sure you would say as a clinician that you don't want people to start smoking?
So this was kind of the same thing, as I said, where we looked at populations of people with Inosine and saw that, higher levels of urate were associated with a lower level of diagnosis of Parkinson's disease. Same thing with smokers. We saw that people who smoked seemed to have a lower risk of diagnosis of Parkinson's disease. So that led scientists to say, is it smoking itself? Is it nicotine? Is it something in the cigarettes that's leading to a lower risk of Parkinson's disease? And that led to the study of nicotine and nicotine patches. And in preclinical models, there was something in the nicotine that seemed to be protective against Parkinson's disease. So nicotine patches went through phase 1 trials and are currently finishing up phase 2 trials. And that data is currently being analyzed, and then hopefully nicotine patches we'll see the results of that early next year. And then nicotine patches will go into phase 3 trials to same thing. This will be a multi-year trial to see if these nicotine patches could slow the progression of disease.
And let's take up one more, Todd, and have you describe Exenatide, the diabetes drug. Again, it seems, as you described with Exenatide, at first it seems like, well, what in the world would a hypertension drug have to do with Parkinson's? You have the same sort of reaction here. What in the world would a diabetes drug have to do with Parkinson's? And yet there is a connection, which right down at the cellular level. So give us a sense of that, why it's intriguing, and what we know about it so far.
Yeah, this is sort of a very similar story where some of the cellular mechanisms that are involved in diabetes have now also been linked to the survival of the brain cells involved in, implicated in Parkinson's. And Exenatide is a molecule that is used to treat diabetes and has that biological function that we're interested in that could have an impact on the survivability of the brain cells involved in Parkinson's. And there have been some very nicely done laboratory studies to show the neuroprotective capability of this particular compound. This project also went through an early clinical test. Again, a lot of these focus very early on safety because, again, most of these, when you're repositioning a drug, the drug was designed originally to be given to somebody who had a very certain type of disease and not given to the broad population. So you do need to do that assessment. And the first clinical trial that was done in Exenatide established a dose and had some interesting potential in terms of some of the clinical signals that we're seeing. The phase 2 trial was recently completed, and those results will be available also, I think, in the first quarter or so of 2017. And based on those, the data that we'll find, we'll see what to do next with this particular drug. This pathway is getting a lot of attention because in addition to Exenatide, there was also a recent publication that was led by Patrick Brunden from the Van Andel Institute looking at other compounds that have a similar type of biological impact, and those seem to also have very promising data in the laboratory. So I think, a lot of these are listed here as individual drugs in terms of isradipine, Exenatide, but there are a number of other compounds moving through the pipeline that have the same biological function. So there's sort of backup opportunities based on how these perform in the clinic.
And we'll talk a little bit more later about those underlying pathways and some of the connections that we now seem to be closer to understanding about the way in which Parkinson's works and the clues that then provides us to figure out ways to intervene. Before we get to nilotinib, the last item on this particular slide, though, I want to ask you, Todd, about a question that just came in from Ken, which is an interesting question about isradipine, this high blood drug that was originally designed for hypertension. And his question is, would it make any sense for someone who doesn't have Parkinson's but is at high risk for it to take isradipine? I don't know whether in Ken's circumstance that has to do with perhaps a genetic mutation or a family history or whatever, but because of course one of the goals would be to stop Parkinson's before it even starts. Is that something that we could conclude once we get through phase 3, or is that an entirely different kind of scientific question, whether it could have that sort of preventative capability?
Yeah, so I guess a couple of different aspects of this question, and I'm going to let Rachel weigh in as well. So first of all, if you are somebody with Parkinson's or at risk for Parkinson's who already needs a hypertension drug, that's sort of one category, and it's probably worth then discussing with your doctor whether, your cardiovascular doctor as well as your PD doctor, whether there's any reason that it would matter if you switch to isradipine versus the other calcium blocker you may be on. So that is sort of one category where if you're already being treated for hypertension, this is a drug that may be appropriate for you. Again, I don't know everyone's individual situation, but something to talk to your doctor about. The other question in terms of what we can sort of extrapolate from a trial with someone with Parkinson's to preventive trials, and I don't think that is particularly known not only for Parkinson's but for other neurodegenerative diseases. But one of the things that is done in just about most of these studies that we're talking about here for the goal of slowing or stopping Parkinson's is that the trials are being conducted in very early stage Parkinson's patients. And the reason for that is really because that is where we would hope to find the largest signal in a clinical trial. It doesn't necessarily mean that once we demonstrate an effect that that's the only population of individuals that could benefit from the drug, but that is what the trials are being conducted in. To actually conduct a formal trial on the prevention of Parkinson's disease at this stage would be incredibly difficult because of our, and we'll talk more about this soon in terms of our ability to develop objective tests and measures of the disease and of at-risk people at risk for the disease. So because we don't really have those great objective measures, that kind of preventive study would require an extremely large number of individuals. So I'm not sure that we would have formal data on it in any time soon. So I think that's a tough question. I don't know exactly that we have data to support it. So I think it sort of thinks about, the categories of what your individual situation could be. And I don't know, Rachel, if you want to add anything to what I...
Well, I think I would just add a little bit of a broader answer to the question we commonly get, which is, should I take this blood pressure medication if I'm already on another blood pressure medication and I have Parkinson's, or should I take this diabetes medication if I have diabetes and I already have Parkinson's? And, as Todd already alluded to, there may be a reason that you're on a specific blood pressure medication. Sometimes doctors prescribe a specific blood pressure medication because that one is protecting your kidneys, or that one is slowing your heart rate, or diabetes medications are indicated for specific reasons. So it's not a bad idea to have that conversation, but asking those specific questions, because you may be on a specific medication for a specific reason. And then when thinking about, these medications that are already out on the market and taking them, when they're not specifically proven to slow the progression of Parkinson's disease, knowing that every medication has specific side effects and thinking about those.
Well, that leads us, I think, into the last item on this list, which is the cancer drug nilotinib, because it ties into this question, which understandably when we hear about something that may have promise, there's an eagerness to take it, and if it's a drug that's already approved, then there's this possibility of picking that drug and sort of getting in on it early, using it in a so-called off-label way for a condition other than the one for which it has been approved. And that question has come up now with nilotinib. So let's spend a moment on that, and I'm eager to get the perspectives of both Gary and Todd on this. First, Todd, on describing what the drug actually is. Again, this question of why would a cancer drug work for Parkinson's? In this case, it seems like it has something to do with the protein mechanism that we think is involved in Parkinson's, and perhaps connected to something called kinases, which are also involved in some of the genetic mutations. So what is this drug? What does it exactly do? Why might it have promise in Parkinson's?
So nilotinib targets, again, another cellular pathway that has implications across diseases. In this case, it is a kinase that's called the c-Abl kinase, and nilotinib acts as an inhibitor of that kinase pathway. What a kinase does is it's sort of a protein that, when activated, tells other proteins within the cell to do certain activities. So it's a switch within the cell that can initiate a cascade of activities within that cell. And just sort of through the knowledge of these diseases, what was found is that the activation of that kinase can lead to certain blood cancers, or be implicated in certain blood cancers, and that's how nilotinib was originally developed. More research over the last few years has implicated that same pathway in the survivability of the brain cells involved in Parkinson's, and the impact of some of we'll talk more about soon, the alpha-synuclein pathology. So there was an initial study that was conducted. Really, again, the main goal of the study was focused on safety. Nilotinib, being that it's developed for cancer, while effective in the cancer, does have certain safety warnings that are part of the instructions for taking the drug. So that was one of the initial study that showed that a small number of people were exposed to the drug, and there was some information garnered about the safety and some initial descriptions of some potential clinical effects. Right now there are a number of additional phase 2 trials that are being planned to try to follow up on that initial observation, to garner more data, to provide more information both to the patient community and the doctor or physician community on what should be done next in terms of this drug in treating Parkinson's.
And Gary Refaloff, you've been involved in this, and that you serve as a patient advisor to one of the trials that's being developed in conjunction with the Michael J. Fox Foundation and a couple of other research institutes. I'm interested in your perspective on this, and in particular, the eagerness with which this news about this particular drug has been greeted within the Parkinson's community, and your thoughts about, because I know you've mentioned in an earlier conversation we had, your awareness of people who are taking this drug in an off-label capacity before we have finished testing on it. What's your sense of what's going on with that, and what are some of your thoughts on that as someone who has been so actively engaged in as a patient participant in clinical trial research?
Well, this is probably a great example of how the press sometimes misleads us, and we're all aware of the headline stories is what gets our eyes when we get news, and this was another example of the research actually came out well over a year ago, although the formal study didn't hit up to this past summer. But when it first came out, it was, cure is found, and it was such notoriety that it excited people, who want excitement, because we're all looking for something that'll slow or stop Parkinson's disease. And the media just picked up on this. It was in every paper, on the news, on television. And unfortunately, not that it's still not exciting, but when the actual information of the study came out, some of the excitement by those who are a little bit more attuned to understanding the research scientifically were not as excited. Not that it shouldn't be something to go forward and get tested in the next phase, which it is, but it's not necessarily, the cure-all. It hopefully will work, but we don't know. And I guess from my opinion, I didn't see it as any more exciting as a lot of the research that is going on this year. It was a great year for Parkinson's research, whether it be vaccines, the monoclonal antibodies, nilotinib, Exenatide. There's a lot of good research going on, and there's a lot of trials that'll be beginning in 2017, and this is one of them. So the good news with nilotinib is that we have 3 separate studies that will be beginning to recruit sometime during 2017. So there'll be a lot of opportunity for volunteers, people that, patients that are interested in taking the drug, to volunteer to get into one of the studies. And that's really what we should do. And to expand upon what Todd said, it's not only that there are warnings with nilotinib as a cancer drug, but there's actually what's called a black box warning on it, and it involves certain heart conditions. So it's not the type of drug that you would want to take off-label on your own, and I'm aware of a lot of people who are doing it. We've probably in our research have come across almost 200 people that have responded to questionnaires that are actually taking it off-label. This is not the type of drug that you'd want to be taking without medical help and advice. Blood tests are required, and heart scans are required. So I certainly don't advise anybody to do it on their own. I could understand why somebody may take isradipine. They're taking a hypertension drug to begin with, and they just switch to another hypertension drug. I could see that certainly more than on your own trying to use a drug that is for leukemia to try to solve a neurological disease, which we don't know that it actually works at this point in time.
And Dr. Rachel Dolan, let me ask you just for one last comment on this, and then we need to move on. But as Gary is suggesting all of these drugs involve potential side effects. We've had a couple of questions already about, well, what side effects are involved with these various things we've been discussing. But this is why it's important to not jump to one of these before it's been fully tested. If you would just say something more about that, and about the fact also that, as both Todd and Gary have suggested, this was a very small trial. It was only, I think, a dozen individuals, not all of whom actually had Parkinson's. Some had other Parkinson's, similar conditions to Parkinson's. So there's just a lot we still don't know.
Yeah, I think it's, as you said, and as Gary and Todd have said, it's just so important to remember that every medication, every supplement has potential side effects, and to discuss everything you're thinking about taking with your provider before you do it. Isradipine can cause swelling of the legs, dizziness. Inosine can cause gout or kidney stones. Exenatide could cause low blood sugar. Nicotine could potentially worsen tremor. Nilotinib, as Gary said, could cause heart problems. So everything comes with potential side effects, but also potential benefits. So that's why we've got to test these drugs in people with Parkinson's, because we need to make sure that they're safe and tolerable and effective.
And of course, again, that doesn't mean that we're not excited about these possibilities, whether it's nilotinib or inosine, isradipine, and all the rest. It's just that we want to make sure that they both have an efficacious benefit for people with Parkinson's, but also that they're safe to do so. All right, let's move on to our next series of topics that have to do with being able to slow or stop Parkinson's. We'll have to pick up our pace a little bit, everyone. We spent a half hour on our first slide, so even though we have 90 minutes, we'll need to keep moving. Let's talk first about alpha-synuclein, which has been so front and center, of course, in Parkinson's research now for many years. The exciting part, Rachel Dolan, is that we now have 5 different ways of trying to confront alpha-synuclein that are in human trials. So this is something where real progress is being made. Each of these are in different stages of testing. They're different approaches, but give us some sense of how these collectively, we hope, will tell us something about a way to stop either the buildup of alpha-synuclein so that it won't form the clumps that we think are crucial to the development of Parkinson's, or can break up those clumps once they're formed.
Yeah, so just to remind everybody, alpha-synuclein is that protein that everybody has, Parkinson's or not, that in people with Parkinson's clumps up in the brain and we think contributes to the death of the brain cells. And so we think by targeting that protein, either preventing those clumps from forming in the first place or clearing out those clumps, we could potentially slow or stop the progression. And so this is a real priority area for therapeutic development for us. And so seeing 5 therapies that work on this pathway in clinical trials is really exciting. And as you mentioned, these 5 therapies work on different parts of that pathway. So just to quickly go through those, one is a vaccine approach. So this is similar to something like a flu vaccine or flu immunization, where we would give a vaccination that actually makes the person's body make antibodies against the alpha-synuclein clumps. So these antibodies go in and actually clear out those alpha-synuclein clumps. And then 2 other companies are working on what we call passive immunotherapy, or they're making actual antibodies outside the body and then infusing those antibodies, which, just like the active immunotherapy or the active vaccination, go in and clear out those alpha-synuclein clumps. Then there's an oral drug which would go in and prevent the clumps from actually happening. And then finally, there's a drug that's in the works that works on just clearing out the misfolded protein. So it's a little bit more of a general approach and could potentially work on multiple diseases that have misfolded proteins. So it could work on things like Alzheimer's or even multiple system atrophy, which have different kind of misfolded proteins like alpha-synuclein and beta amyloid. So again, varied approaches on different parts of this pathway that are targeting alpha-synuclein.
And Todd, we can see that these are not quite as far along as some of those repurposed therapies that we were discussing a moment ago. These are now just kind of entering the phase 2 part of this. So we're further away. But give us some sense of I guess, the overall focus and excitement of the scientific community in this arena, because there have been some exciting developments in the Alzheimer's area that have told us that this really may be something that can work. Could just sort of summarize, I guess, the why this is so central to our pursuit of finding a cure for Parkinson's and what the sort of mood or sense is among the scientific community about where this can go.
Yeah, so just 2 comments. One first, while these definitely are sort of entering the phase 2, so they might be behind some of the ones on the other slide. They actually are ahead of some of the other ones. So some of these, for example, are ahead of nilotinib in terms of the stage. But also just to remind everyone that while it says in parentheses 5 in trials, you would see 5 lines, not 1 line, if you thought of each of them as you compare it to the slide before. The reason why I think people are so excited about this is that these trials are really the culmination now of probably a decade of laboratory research that was really focusing on how can we increase our knowledge of the underlying triggers and biological pathways that are responsible for Parkinson's. And I think in this area we're really trying to develop therapies to slow or stop the disease. The therapies that are most in line with what is the underlying biology, the underlying cause of the disease, have the greatest chance of success, because that's really what we want to target. We're trying to move away from just replacing the dopamine that's lost in the disease. We're actually trying to prevent the process that is leading to those cells to degenerate in the first place. And right now I think that the alpha-synuclein role in that process is really central, and it's really centrally believed throughout the scientific community to play a very important role in either the trigger or the progression of the disease. So the fact that now, based on that significant laboratory data, that we have a number of these trials that have gone through that true translation process and conversion to clinical testing is an important area of optimism, in that these are really some of the first trials targeting this particular pathological mechanism of the disease.
And Todd, would that also then be the case for the next 2 things we see on this slide, the GBA and LRRK2? Those are both mutations that are associated with either increased risk of Parkinson's or actually leading to Parkinson's. They too have been teaching us something about these underlying pathways that you've been describing. How close are we in either of these cases to actually starting trials in individuals that would counteract the impact of some of those gene mutations?
Yeah, so that's exactly true. That one of the key features that we look for the biology we want to target for therapy is knowing that this biology is altered in some way with the disease. And one of the leading ways of doing that has come from genetic research, and both the GBA and LRRK2 mutations have been identified as critical players in Parkinson's. So both of these are moving quickly. The GBA is a little further ahead in terms of the clinical development, and I would anticipate within the next 3 to 6 months, if not sooner, a trial being launched in that area. LRRK2 probably within the next 12 to 18 months, having a therapy targeting the particular mutation. And what's interesting about this too is you probably have heard a lot in the news and in areas like cancer of this more personalized medicine approach, where in cancer, for example, they will look at the tumor under the microscope and do testing and can say you have a cancer marked by this particular genetic mutation, and then they can come in with a more targeted therapy where they know that this type of therapy can have a chance for a better success with that molecular characterization of that tumor. These examples, particularly with the GBA and the LRRK2, are sort of the leading edge where we're starting to get in this direction in Parkinson's. We've talked many times on these webinars, and I'm sure you've all heard this before, about how heterogeneous Parkinson's is, how variable it is. And now we're starting to make more sense of that variability and have this capability of perhaps having more targeted therapies. And one thing we'll talk about next, I think, is also how having these leading edges of targeting these genetic mutations is also uncovering the possibility that some of these therapies may be more widespread and have greater impact for people, even those who don't have these mutations. So this is incredibly exciting and a new age of Parkinson's research, a new potential for therapies to target the underlying cause and progression of the disease.
And before we get to that, Gary, I just want to ask you what you were talking about before as far as opportunities to participate in trials. If you're someone who's interested in participating in any of these things, in alpha-synuclein and also when both of these genetic drugs go into trial, these are great opportunities, right? Because recruitment is either just beginning or hasn't even begun yet. So these are things really people can keep an eye on.
Yeah, and as Todd said, so many of them are going to be recruiting over the next year for trials that are starting. So it's not only the nilotinib that we were talking about, but at least 3 of the vaccine monoclonal antibodies that Rachel was talking about. At least 3 of them are going to begin, and possibly more. So I think the opportunity right now is there for us, as patients, to do 2 things. One, to get ourselves potentially into a trial that has a treatment that may work in slowing the progression, and 2, get ourselves into a trial that is going to help move the science of Parkinson's disease forward, which is one of our obligations as patients, and 3, doing it through Fox Trial Finder by staying on top of it. The faster we volunteer and get into these studies, the shorter the time period will be from start to finish. And it's unfortunate that a lot of the delays of some of these studies is because the patients are not volunteering. So I think it's a great opportunity, and I'm very excited about the upcoming year with studies that you've been mentioning.
Yeah, I think it's a good example of what is sometimes referred to as enlightened self-interest. I mean, we have a real self-interest in participating in these trials because it may do us good, but it also advances the science, and in that way can help enlighten the broader field of scientific research. And no one can do that better than those of us who have a Parkinson's diagnosis. Let's move to where Todd was just describing, which is what all of this is beginning to tell us. And we see at the top of this slide sort of connecting the dots between Parkinson's biology and symptoms. And we're putting together here everything from alpha-synuclein to the LRRK2 mutations that we were just talking about, to GBA, and beginning to make some linkages. And Todd, this is something that I find as a patient incredibly both interesting and exciting, because at least it feels to me, and I'd like you to give us something more objective than just my feeling, that we really are beginning to make connections here. We've been talking about the role of proteins that clump together and the underlying biology between that. We know that some of these things involve cellular mechanisms like the mitochondria. We now have some drugs that may work on that, and that this has to do perhaps with helping the cell get rid of the bad stuff. And all of these things begin to link together, the relationship between some of these genetic mutations like LRRK2 and alpha-synuclein. Is my feeling justified, I guess, is where I want to go first with this. Are we really beginning to connect the dots?
Yeah, this is probably the most exciting area in terms of the laboratory research right now for Parkinson's, and I think it has great impact and implications for the future of therapy. So as we started to discover over the last 5 to 10 years these various genetic mutations or genetic links to Parkinson's, it started initially to look a little just like a scatter plot, where we would find this subset of patients that may have this mutation, this subset of patients that may have this mutation, and a large set that didn't have any mutation. But it was hard to make sense of how is this all connected, how does this ultimately lead, for example, to the commonalities of the disease, the loss of dopaminergic function, the motor symptoms. And as we've now been able to study more and more the impact of these individual genetic mutations on the brain cells and other cells within the body, it's starting to highlight commonalities in terms of some of the biological pathways that could be leading to Parkinson's disease, even if the initial trigger of that process, whether it be a genetic mutation or an environmental exposure, may be different. And this is really important because it actually expands the potential for the impact of the therapies that are being developed now. So, for example, if you're developing a LRRK2 inhibitor, initially we may have thought that that inhibitor would only be effective in someone that had the LRRK2 mutation. But as we've now uncovered different cellular pathways that may be altered in others with Parkinson's who don't have the mutation, you can envision that perhaps that LRRK2-based drug could impact a larger percentage of the population. What this also has done is indicated completely new outside-the-box approaches that we can go down to develop therapies, as we've uncovered more of the impact, for example, like you said, on mitochondria. Or another area that has come out of this biology involves neuroinflammation, which can have a broad impact across the population and leading to, impacting the progression of the disease. So I think this is an area of incredible importance and, highlighting a lot of the innovative research that's been going on over the last few years in the Parkinson's community. The last point that I would make on this, which I think I'm going to talk a little bit about in the future on this call, is that what this also is uncovering is different ways that we could go about developing blood tests or other types of tests to analyze Parkinson's disease as we really start to understand what the biological and cellular impact of these genetic influencers are on the disease.
And that's, of course, crucial, as someone was just asking a question about, well, how are we going to measure whether or not some of these therapies work? And that's the big question, right? We need to be able to have objective measurements. Otherwise, we won't know whether or not these new approaches definitively work or not.
Yeah, that's exactly one of the biggest challenges I think we have now in Parkinson's research, because while the therapeutic pipelines are becoming more and more innovative and more robust, we still have this common challenge of really diagnosing the disease as early as possible and as definitively as possible, and then having good measures of sort of tracking the stage of the disease and the progression of the disease, so we know whether any of these drugs have actually slowed that progression. And that's a major area now of research, as you know, that the foundation and the field is really focused on.
And Rachel, one of the things that's mentioned here on this slide too, I'd like your perspective on, which is the connection between biology and symptoms. Todd was talking a moment ago about we may be getting closer to a time when we can apply a more personalized or precision medicine kind of approach to Parkinson's, because we know Parkinson's varies so much. What may treat Gary's Parkinson's best may be different than what treats mine best. And we see on here a couple of specific connections here, that people who have the LRRK2 mutation might get Parkinson's sooner. People with a GBA mutation might have greater cognitive decline. That's part of why I assume this is so important too, is to tailor approaches that will connect specifically to the symptoms that people have that may be the most problematic for them.
Yeah, it'll help us tailor approaches, but it'll also help us with prognosis and therapeutic development. So I think it's so helpful, and also, shows us how much we're learning and, how advanced we're getting. It says on the slide, people with a LRRK2 mutation and another genetic mutation may develop Parkinson's sooner, but we also know that people with just the LRRK2 mutation actually look like people with just regular old Parkinson's, people with Parkinson's that don't have a genetic mutation. So that tells us a lot, like Todd was just saying, that actually the therapies that we're developing against LRRK2 may be widely applicable to the broad population of people with Parkinson's who don't have a genetic connection. And then, as you pointed out here, people with Parkinson's and a GBA mutation may be more likely to have cognitive problems or memory and thinking problems. So that may help us, develop therapies against that, or design our trials better, or help them with their planning, just in life. So all of these things are, I think, helping us clinically with people with living with Parkinson's, but also helping our research.
Let's spend a little bit of time next on advances in symptomatic therapies during 2016, because there have been some important improvements in this arena too. And this is crucial because, of course, while the end goal is to figure out a way to slow or stop disease progression, in the meantime, we have to live with Parkinson's. And so having better symptomatic treatment in the here and now is crucial as well. And we see a number of things, and we won't go through all of them in detail, but in 2016, a new drug was approved to deal with hallucinations and delusions in Parkinson's, which is a problem that had been untreated. We also see on here some things that are going to be close to winning approval in the next year that have to do with better treatment of both dyskinesia and on-off episodes. As you look at this, and then Gary, I'm going to want to bring you back into this conversation. As you look at these symptomatic improvements, Todd, what stands out to you in terms of the focus on this? For example, with on-off, this is something where we're now really, this is a big problem, obviously, for people living with Parkinson's, and we're close now to really being able to contend with that. How important is that we've made those sorts of advances?
Was that to me?
Yes, sorry, Todd, to you.
I'm sorry. Well, clearly, we've focused so much on the holy grail of slowing or stopping Parkinson's that we, lose some of the excitement when there's advancement in symptomatic therapies. And we are very close to having 2 drugs approved that will very much help control on and off symptoms. The ease of taking an apomorphine sublingual strip or potentially an inhaled formulation of levodopa will improve life greatly for those who have the bad off periods. And there's no doubt that it's one of the symptoms that is there for most of us that doesn't get discussed all that often, but many just live in fear of when that drug wears off. How do we get to the next dose and survive during that time period? So it's very exciting, some of the advances that have happened with some of these symptomatic therapies, including potential drugs for levodopa-induced dyskinesia that could happen relatively soon. And, just from the patient's point of view, I think there are many of us who focus so much, as I said, on the interventional cures, but there's millions of people living every day with Parkinson's who would just be happy if they can get something to relieve constipation, and they're more focused on that as opposed to, nilotinib or vaccines or antibodies.
Exactly, and I know you particularly feel that we need to bring more attention to some of those non-motor symptoms that are listed at the bottom of the slide. And if we're going to start taking all of your questions in a moment, and so if people have questions about any of these particular new therapies that are either on the market now or will be on the market soon, do get those questions to this. I think we'll not spend more time on this now. Just we have so much to still get through in our hour-and-a-half webinar today, but if you do have specific questions about any of these particular new medications, get those to us and we'll be responsive to them. Let me just move us to our next area, which is the thing that we've alluded to a couple of times but haven't really spelled out yet, and that's getting closer to an actual test for Parkinson's disease. Not only, of course, because we want to be able to diagnose sooner, but also, as Todd has alluded to before, we need ways of objectively measuring whether or not these new therapies that we're so excited about actually work. And we see a lot of different things here on the screen, and I'm not going to take time to discuss all of them, but I'm just going to ask you, Todd, to zero in on where you think we're getting the closest to be able to make those objective determinations. And maybe it's several, maybe it's in the spinal fluid, but it's also in developing a scan where you could actually see alpha-synuclein in the brain. What to you is the most promising in this general arena of being able to develop more objective tests?
Yeah, I think the most important area really is trying to develop better brain scanning neuroimaging approaches to see what's actually happening in the brain of people with Parkinson's. Unlike I keep making a reference back to cancer where they could take the tumor out and look at it under a microscope. Unfortunately, that wouldn't be a very feasible approach when the disease is in the brain. So we have to go one step away, which is using neuroimaging. And it would be an incredible advance to have the ability to image the alpha-synuclein aggregates in the brain to then be able to tell whether any of the treatments are resolving those aggregates. And there has been significant progress in that area, although there have certainly been some technical difficulties. But we can see in the Alzheimer's space how having an imaging scan that could look at the aggregates that are impacted in Alzheimer's has had a significant impact in really advancing the therapeutic opportunities in Alzheimer's disease. I think one of the other areas that I just wanted to highlight is looking at other technologies. One of the things many of you are probably familiar with is how non-precise or imprecise the current clinical exam is for Parkinson's disease to really get a fine-tuned evaluation of the symptoms and the impact of those symptoms. So there's been a lot of work in the last year or last few years to both try to use technology to get more objective measures of symptoms like tremor, like activity, like speed of movement, that can utilize a lot of the devices that exist now where people use these for exercise or various watches or things like that, or keyboard tests that could be given more remotely to get more objective and consistent measures of the disease. And also to look at some of the other symptoms that are impacted in the disease. On the slide we talk about eye movements, but there are a lot of these non-motor symptoms that also are not impacted by the current medicine as well that can be used to get a more objective measure of some of the symptoms of the disease. This area could have, profound impact. This would really change the playing field on which we go about developing therapies, and it's an area of great interest and pretty robust research, but we haven't really gotten to the real fine objective test. There is available a dopamine scan that can be very effective for helping with diagnosis, and the data from our Progression Markers Initiative is actually showing that test itself can be useful as well to track some of the early progression of the disease.
And Gary, you're someone who has participated in that Progression Markers Initiative test, and I also know you've participated in a number of wearable studies. Todd was just mentioning how we're all familiar with how imprecise diagnostic is with Parkinson's. We're all familiar with going into our neurologist and having that person, have us tap our fingers and stomp our feet, and it's hardly the most objective test in the world. If you would, Gary, describe a little bit about how wearables can help with more detection of what someone's symptoms are, because after all, we only see our neurologists for an hour or two a year. And wearables have the advantage of collecting data 24/7. How might that also improve our ability to both detect and monitor what's going on with someone's disease progression?
Well, something that's gathering information about our symptoms 24/7. And if we use it correctly, we may be able to tie in individually certain conditions, how we take our medications, when we take our medications, how it may impact tremor or dyskinesias. So it very well can lead to a much more individualized treatment for us, beyond the scope of just getting research information from a large group of people. If and when we can get it down to the science of using a wearable device individually that supplies information to our doctors, so when we go to a quarterly or semi-annual visit, they can actually look up and they'll have information. And when they ask the question, as an example, how often are you exercising? And we say, well, every day. But then when they look at the data and it doesn't look like we actually had 30 to 60 minutes of activity, maybe we really aren't exercising every day. So I see the future of us being able to monitor ourselves individually in terms of our medications and activity, and tying it into trying to control our symptoms.
It's a nice way to put it, in that it's something that can both help us monitor our own experience of Parkinson's as well as provide that data to clinicians.
Exactly.
I'm going to put up on the screen here a number of other things that have come up and that we want to discuss, but I also want to just start folding in the questions that we've gotten throughout our hour plus so far, and use this last 25 minutes to not only mention these items that are on the screen, but a number of other questions as well. So let's start with a question that's come in about deep brain stimulation. Someone raised the question about whether or not DBS can be effective for non-tremor dominant Parkinson's. Rachel Dolan, why don't I put that one to you? We know that DBS has been particularly effective where tremor is a problem, less effective with some of the other symptoms of Parkinson's. So how are we trying to expand, I guess, the repertoire of what DBS can effectively treat?
So to answer the question, typically we say that DBS is most effective for the symptoms that respond to medication. So that's going to be usually the stiffness, the slowness, the tremor, less commonly the walking and the balance problems. It can be effective for tremor that is resistant to medication or doesn't respond to medication. Now, for walking and balance problems, there is a lot of research ongoing into determining whether we can target different areas of the brain that could respond to deep brain stimulation. There's also research into looking at, could we program the settings differently, change the frequency or how we deliver the electrical stimulation to help with the walking and the balance problems. There's also research into, can we personalize deep brain stimulation? So like Todd was talking about, just personalizing medicine into kind of these genetic targets. Can we personalize deep brain stimulation? Can we deliver the electrical stimulation on demand when it's needed for a person's kind of unique electrical signals in their brain? So a lot of research into expanding deep brain stimulation for not only the symptoms that it would be applicable for, but the people and the population that it would be applicable for.
And Rachel, when it says under our slide here, deep brain stimulation, a new system approved in 2016, is that a new system that does things differently, or is that just like a different car model that's now available for people to choose from?
Probably a little bit of both. So there is, previous to this system, there was kind of one system that was approved since 2002, and that was the Medtronic system, which had a couple different models. And then earlier this year, one called the St. Jude Infinity model was approved. And it's kind of the same component, so it has the wires that are implanted into the brain, and then that neurostimulator, which is in the chest, which is what's programmed to deliver the electrical stimulation into the brain through the wires that are implanted into the brain. What's a little bit different about this new model is that the electrical stimulation that's delivered into the brain can be sort of steered toward the target of interest. So when we say that, it can be a little bit more honed in to where we want the electrical stimulation to be delivered. And that can mean that it can be more honed in to lessen the Parkinson symptoms and potentially avoid side effects. And although the current models or the previous models, the electrical stimulation is delivered over a very small and specific area, this is even more specific.
And on moving to stem cells, Todd, a number of people are asking where we're at with that. This is a subject that seems to kind of wax and wane in terms of how much scientific focus there is on that. It's referenced actually in the new 21st Century Cures Act that the President signed the other day about regenerative medicine. Are we making any real progress towards having stem cells be an effective source of therapy for Parkinson's?
Yeah, this is an area that over the last 12 to 15 years has had its sort of peaks and valleys, but I'm pretty excited right now that we are on an upswing of where this research has gone. So the goal here is to try to take stem cells and convert them into the dopamine-producing cells that are lost in the disease, and then ultimately transplant those back into the brain to replace the cells that have been lost. It's pretty well established now that you can, out of human embryonic stem cells, make very high-quality dopamine-producing cells. There had been some challenges in the past about then getting that transplant to work and function and survive in animal studies on the way, trying to build this towards the clinic. My sense is that over the last couple of years, many of those challenges have now been overcome, and we're now sort of back on the upswing where I'm aware of a number of groups that are kind of conducting some of the last stages of research before launching new clinical trials. And it would not be out of the realm of possibility that within the next 12 months or even 12 to 18 months we would have some new trials being launched in this area of converting these stem cells into dopamine-producing cells. There is another area where there's been a major advance in the last few years with stem cells, which is in an area called induced pluripotent stem cells. This is a technology that came out of the original research on human embryonic cells, where you can take an adult cell, like a skin cell or a blood cell from somebody who's with a disease like Parkinson's, and then using these genetic and other cell culture technologies to convert that cell back into a cell that has more stem cell-like properties, which means it has the capability to be engineered into other cell types like a brain cell. And that technology is being used really in two areas. One is in some ways to develop a disease in a dish. So if you take a cell from someone with Parkinson's in the laboratory, now convert that into a brain cell, you can study the cell and understand what might be different in that particular cell versus someone without Parkinson's to understand the biology and develop potential therapies. But there also can be used in the same approach around transplants, where you can take that induced pluripotent stem cell, convert it into a dopamine cell, and consider it for transplant back into the individual, overcoming some of the potential immunological limitations of transplant because you've taken that cell from either the individual or someone with a similar immune characteristics as the individual is getting the transplant. So this is all really almost science fiction, but it's not. It's actually happening and really exciting. One caveat I would raise, though, in terms of the stem cell therapies is that right now the main focus of these is to replace the dopamine cells that are lost in the brain. So as we know, there are many symptoms that people have with Parkinson's that don't respond to the dopamine-based therapies. So it's probably going to be a similar case where if the stem cell therapy transplant occurs, you hopefully would impact a lot of the dopamine-dependent symptoms, but symptoms that are not related to that particular brain region or to the dopamine system may not be as impacted by the therapy. The last caveat I would raise about this is an area where I think we want to be really careful and really rigorous in the assessment of the underlying data and process through which an individual stem cell therapy has been brought forward. For many years now, there have been these stem cell clinics, both in the U.S. and overseas, that have claimed great results, but without really any solid underlying data to support those results, and that's something I would certainly keep a cautious eye towards as you see that information.
Yeah, I think the rule of thumb there is if you see something advertised on the internet as they offer a stem cell cure for Parkinson's, that means don't go there. All right, let's take up lots of topics coming in. We will get to exercise, but one of the questions that's come in the most today has to do with gut bacteria. There's been some interesting studies that have come out recently that seem to show that people with Parkinson's may have something going on differently in the gut with their bacteria, and that somehow has a connection to what's going on with Parkinson's. Rachel or Todd, either of you, can you explain a little bit about what we're learning about that and what's intriguing about it?
Yeah, so this in my opinion, is extremely interesting. And for one reason, we talk a lot about what causes Parkinson's, and in many areas, I've done these types of presentations for many years, and I'll give an answer that says it is the interaction between your genetics and your environment, which is another way of my saying I have no idea what causes Parkinson's, because what isn't the interaction between your genetics and environment. But we've actually learned in recent years that there are certain parts of your body that may be impacted initially in Parkinson's disease, even before some of the motor symptoms are identified. Two of these include the sense of smell, your olfactory system, and the other is your digestive system. There was a study a number of years ago, an epidemiological study that showed that the symptom of constipation can predate the diagnosis of Parkinson's by 5 or 10 years. What's interesting about both of those systems, the olfactory system and your digestive system, is that is actually in practice where your genetic makeup directly interacts with your environmental exposures, whether it's through something that you've inhaled or breathed or something you've eaten. And what is interesting about this recent study is actually there's now developed some evidence suggesting that the microbiome, or your sort of the bacterial components of the digestive system, may differ between people with Parkinson's and those without Parkinson's. So that observation itself wasn't that new. There had been some studies on that in the past. What was the kind of most recent step forward in the recent study was then trying to take that observation a step forward from the correlation, which means this is just different between someone with Parkinson's versus someone without, which could be caused by a lot of reasons, to try to see whether there was actually something pathologically with a causation aspect of that microbiome. And they did that by transplanting the microbiome from someone with Parkinson's into an animal model system, and showed that actually when you did that transplant, the animal showed more Parkinsonism, to the extent that an animal mouse shows that. So that indicated that maybe there was actually something within that microbiome that could somehow be contributing to the pathological process in Parkinson's. So I think this is really interesting. for all the reasons that I just stated, and sort of opens a whole new opportunity of figuring out if we can understand better what might be going on with that microbiome. Is there a way that we could either correct it or use that as a way to develop new therapies or give better recommendations around diet or things like that? I think that is sort of where the next phase of this could go. And I guess I should have stated before going through that whole dissertation that for those that don't know, while we live in a world, we ourselves are a world, and there are microbes and bacteria and things that live within our, in our body, and it actually is, in many ways good for us. It helps us digest food in this case, or handle some things that we're exposed to. So that in that case, what's being seen here is that the makeup of that bacteria is different in the digestive system of someone with Parkinson's versus someone without. These studies are actually fairly complex to do because you need to have very good control systems because, for example, if my wife and I would have by nature a similar microbiome if we eat the same things. So you need to really understand how you're doing the comparisons when you say that someone with Parkinson's is different than someone with controls, and that's where I think some of the follow-up studies will have to go.
And part of what's intriguing about this too is it gives, I think you're suggesting, Todd, another target and a place actually where other drugs could go. And it may be easier to get drugs into the gut than it is sometimes to get drugs into the brain. So a lot of intriguing possibilities may come from this. Let's go through a couple of things quickly, if we can, that I'm going to turn to you for, Rachel Dolan. Someone raised the question about statins, drugs used for high cholesterol, perhaps making a linkage to drugs that are the hypertension drugs that we're now testing in Parkinson's. Anything you're aware of in terms of any problems with people who may be taking statins or perhaps should take statins if they also have Parkinson's?
Yeah, I'll just jump in a little bit on this because this has been an area where I think the data has been contradictory in terms of the epidemiology on whether it's actually does in fact decrease your risk of Parkinson's. For example, if you're exposed to statins, there's been similar data in Alzheimer's. There have been a number of trials to look at the impact of statins on people with Parkinson's as well as Alzheimer's, and those results have been mostly non-compelling in terms of whether once you have the disease, would statins impact the progression of the disease. We had funded a study some years ago looking at the impact of statins on the onset of dyskinesia, and that trial was also negative. So there has been data in the literature both showing that statins could both increase or decrease your risk of certain diseases. So I guess based on my knowledge of the literature, I would sort of use the statin as necessary for its intended use in your own health care, and rather than making a decision based on a neurological or neurodegenerative aspect. But I don't know if this ever came up in your clinic or anything, Rachel.
No, I don't really have anything further to add.
Okay, let's switch and go to exercise and then also weight loss. Questions have come in about both. Gary, you're someone who I know has maintained an active exercise regimen. People always want to know what the best exercise is. I don't think we know the answer to that. The answer seems to be just exercise, and whatever you do that will keep you doing that is what you ought to do. But describe briefly, if you would, your own experience with that and your observations of others in terms of why it's important from a patient point of view.
Well, I know it's probably redundant to a lot of patients that are listening in, but there's no doubt that multiple research studies have shown that people with Parkinson's that are active and exercise on a regular basis do better than those who don't. It's probably the only thing that we have under our control to help, control the symptoms and course of the disease at this point in time. So I do get asked the question often, which is the best form of exercise, and I guess the best answer is the silly one that we all hear is it's the one that you do. And it's not great to sign up for a gym and then not go, or to buy a bike and not use it. So I think it's important that people find forms of exercise that they like and entertains them, because they're more likely to do it on a regular basis. And, I don't think we're promoting that somebody should go and do something that they're not capable of doing. And, working with a therapist or a trainer is a great way to do something within the realms of your capabilities. But to the degree you can be on the more strenuous side and do it longer rather than shorter, you're more likely to get more benefit out of it. I'm aware of a number of studies that are actually ongoing right now that are trying to test out differences between forms of exercise. But from my experience and the people that I know, whether it be boxing or Tai Chi or dance or biking, if you're doing it on a regular basis, it's likely to be working.
Rachel Dolan, add to that if you can about anything more we know scientifically, but then touch on this question of weight loss, which is a couple of people have raised, which can seem sometimes like, well, at least there's one benefit from Parkinson's, but it's a mysterious one, right? We don't really know why it is that many people with Parkinson's do in fact lose weight.
So Gary touched on the majority of what I would have said about exercise. I mean, it's just, we know it's so important to exercise regularly. It's so beneficial for so many symptoms, both motor and non-motor. It can help constipation, low blood pressure, mood, sleep. So it's just so important to do exercise. He said exactly what I would have said, which is pick the exercise that you will do, that you feel safe doing, and every form of exercise can be beneficial, whether that's swimming, gardening, puttering around the house. So, just work with your doctor and your physical therapist to find something that will work for you. There are plenty of exercise studies that are ongoing to determine, which forms of exercise can be most beneficial for certain types of symptoms, and those include exercise that could help everything from, like I said, sleep to cognition to certain forms of motor symptoms like tremor. So those are other ways to get involved in research as well. As far as weight loss, that's certainly not a universal symptom of Parkinson's, and that can happen for a variety of reasons, everything from something like smell loss, which can impact your ability to taste and therefore want to eat, and could lead to weight loss. There can also be swallowing problems that can happen with advancing Parkinson's disease and impact your ability to eat, which could lead to weight loss. And even if, dyskinesia is very severe, those can be those abnormal involuntary movements that can happen with advancing Parkinson's disease, and those can lead to, some involuntary forms of exercise that can lead to extra calorie expenditure that can lead to weight loss. So there can be a variety of reasons that weight loss can happen in Parkinson's.
As always, lots of questions coming in. Todd Scherer or Rachel, you could take this one too on medical marijuana. There is reason to want to investigate it, but are we any closer to having an answer to whether or not medicinal marijuana can be useful for people with Parkinson's? Todd?
So, as you said, there is a lot of preclinical evidence that medical marijuana could be beneficial in Parkinson's, but to date, the research has been conflicting. So, some studies say it's beneficial, some studies say it's not beneficial, some studies say we're not sure. I think there are several reasons why it's been difficult to study. So, first of all, medical marijuana is a Schedule I drug. Second of all, there are many different formulations of it. So it can be smoked, it can be used in different oil formulations. So the studies aren't kind of consistent on the formulations that are studied. So it's hard to kind of compare across the studies. The studies that have been done are also on small numbers of people. So it's hard to get good powered studies or large numbers of people who we can report back on. As far as the data to date, it's been studied for tremor and also for levodopa-induced dyskinesias. And again, the data on that is conflicting. So we don't have good reports on whether it is beneficial. And most people would say right now we're really not sure on whether it's beneficial. As far as other symptoms, the non-motor symptoms of Parkinson's, sleep, anxiety, other mood symptoms, there are small reports that it could potentially be beneficial for those symptoms. And then, of course, anecdotal reports abound that it is beneficial for individual people on multiple symptoms. So, at this point, we don't have the data on safety, on tolerability, and on efficacy, and certainly it deserves further study. I think, again, as you said, there is reason to believe it could be beneficial, but we just don't have the data to date.
I want to get to two more questions before we have to wrap up that are kind of grouping concerns that have come in. A number of people, Todd, have raised the question about whether they should get genetic testing. We've talked throughout our webinar today and in other webinars in the past about the intriguing information that we're learning through the genetics of Parkinson's. What's the best advice we can give people about whether or not, if they have a sense that Parkinson's may be something that runs in their family, whether or not they should necessarily get genetically tested?
Yeah, and I'll give my sort of more research side answer on this and then see if Rachel wants to jump in. But right now, with our current treatments, nothing would really change about your current treatment regimen with added knowledge about your genetics. So that, I think, is just where we are currently with the treatments. A lot of the things we talked about were more in the research realm in the future. So I do think, though, that the only way we've learned about any of this genetics was because people participated in that kind of research and trying to understand not only finding the genes, but how they impact the symptomology. And there right now are a number of significant research efforts, including the Parkinson's Progression Markers Initiative and others, where participating in that type of genetic research is having a dramatic impact on the future development of therapies and knowledge about the disease. So I think that's sort of where I would come down. Now, you can find out the genetic testing. There's ways to do that if you discuss it with your clinician. If you do know you have a family history of the disease, you're not going to get so much more information by uncovering the specific gene or not. But some people like to know, and some people don't. So, I know Dave's had an experience on this himself in his own life that maybe he could provide am I allowed to call on the moderator? But maybe he could provide, more of a personal view on this, because it is more of a personal decision at this stage. It's not necessarily going to provide, that impact to direct your treatment.
You definitely can, but I'm going to duck because we're out of time. But I think the bottom line takeaway on that is that whether or not you choose to get genetically tested is, as Todd suggests, I think a very personal decision, and it won't at this point necessarily impact the kind of treatment you have. That said, as Todd, I want to underline what Todd said, that there is enormous value to participating in research, and you can actually participate in genetic research without necessarily learning what your mutation may be. Just participate because you do have a family history. So there's great contributions to be made whether or not you choose to learn the answer or not. With that, I'm afraid we must conclude. Even an hour and a half isn't enough, apparently, when we're doing these webinars to get through everything. I do want to thank our panelists for their participation. To Gary Rafaloff in particular. Gary, thanks so much for being part of our discussion today. And also to our Fox Foundation duo of Dr. Rachel Dolan and Todd Scherer, our CEO. And I would be remiss if I didn't also thank Maggie McGuire-Poole, who is the person who is the the brains behind putting on these webinars all year long, and her colleague, Elise False, for their great efforts in making this series such an ongoing success. So with that, we'll wrap up. We also want to say that we'll be sending a link to the webinar so that you can listen again and share it with others. We will be continuing our Third Thursday webinar throughout 2017, and we'll be announcing very soon what our topics will be in the first few months. So keep an eye on your email for that information. Thank you again, all of you, for participating in these webinars and also for your support of ongoing Parkinson's research. I'm Dave Iverson. Thanks for joining us.